the last half an hour to discussing chemosensing at the highest level, at the level of true organisms, metazoans, and especially insects, which are sort of a prototype for efficient olfactory communication and detection. So by now you should have sort of understood the pattern of these lectures. So we started from the macroscopic scale, describing bacteria as populations, so from the scale of millimeter to centimeter. Then we took a dive and we moved to the level of single cells, so moving 3 orders of magnitudes down in size, from millimeters to microns, the size of a single cell. And then we took a further dive inside the cell to understand the molecular mechanism. And again, we're moving to the scale of the nanometer now, the scale of these molecular complexes and chemical reactions. At each of these levels of description, we've been using a different physics, actually. So at the level of populations, we use equations that describe how the population as a whole evolves in space using number density, which is justified by the fact that we have billions of bacteria in 1 centimeter cube. So it's a large density. We can treat this as a continuum of bacteria, and we use actually formalism like the one in Keller-Segel equations, partial differential equation, right? We build up a physical description, a model at the level we are, right? We are at the macroscopic level, we build a macroscopic model. Then we move at the size of the cell, and we use a different physics. We talk about hydrodynamics of viscous fluids, we talk about diffusion. Again, we account for the physics that is present at that scale and neglect the other effects. And then we move at the size of a single cell, and we have transduction networks, and we write down differential equations or master equations depending on actually the number of copies of your particles, of your molecules that are involved. One description may be more appropriate than the other. They're all part of the same idea. And then you use this new description to describe this new phenomenon at these scales, right? So in the first few minutes, I will just complete this molecular part in a couple of slides, discussing the one of these three aspects that are present in the transduction, that is adaptation, which I haven't discussed yet. And then the rest of the part devoted to bacteria will be meant to understand how to connect the levels. So can we give a theoretical description of the population level starting from the molecular information? So is it possible to go from one step to another? This is something which we know it's possible in physics in general. So we will try to do something which is very similar to going from the kinetic theory to the thermodynamics, if you wish, or to the fluid dynamics. Starting from microscopic information, mean-free path, etc., we go to microscopic observables such as viscosity and thermal diffusivity. This sort of step process going from microscopic to macroscopic is something that helps you clarify things, and I think it's one of the big things that physics always tries to do. Describe properly a model at one level and then understand the relation between different models at different levels. And then if you succeed to do so, you sort of gave a more fundamental description of what is happening at the larger scales, right? So that's very, very coarse philosophy, not particularly exotic. So as I first said, let's now, we have discussed this transduction pathway. We have especially emphasized the fact that their receptors cluster together, forming structures which are actually, receptors are dimers. They form trimers of dimers, as these trimers of dimers are arranged in hexagonal patterns, which are functionally related in sizes of, say, 18 typically 18 receptors together form one functional unit which responds as a whole. It is what is called an allosteric cluster. It's something that switches on and off in a dependent binding, depending on ligand binding, and works as a signaling unit, large signaling unit. And the advantage of having this large signaling unit is that the response of the kinases here, the activity of the receptor, has this form, which is a form which is pretty steep in a range of concentrations. So as I was drawing yesterday, there are these two typical concentrations, K on and K off, which come out from this allosteric switching. Below K off, you basically don't respond. Above, you are saturated. And in between, the response goes like a steep power law. Actually, depends, like the ligand concentration to the power n, when n is the number of receptors that you have in your cluster. So that's an effective way to amplify small differences. Of course, there is drawback. That is, when you amplify differences, you also amplify noise, right? So there should be a trade-off in the level of amplification between how much you want to amplify your signal and you don't want your noise to bring you away from the region where you measure, right? So may I just put the blackboard on? Thank you. So, and a similar thing occurs at the level of the motor. Again, amplification. Here the mechanism is slightly different because there is a spatial component. This conformational spread model tells you that the motor doesn't behave as a single unit, but rather it's made of several units, which, however, talk to each other. And every single unit, which might see is actually a single protein, which makes this ring of proteins. Every single protein tries to convince its neighbors to get active if it's active, okay? And this spreads the activation throughout the ring. So both these mechanisms actually, as I was said, provide an amplification in the sense that you have a signal which for the receptor is the outer concentration of ligand. For the motor is the inside concentration of this KY phosphorylated protein. But you might think of the motor as a receptor for its internal state. It's a sort of proprioception. I respond depending on what I'm feeling inside of myself. That happens also in our cells all the time. So the signal elicits a response. And this response might be the activity of the receptor or the switching rate of the motor. And typical curve are just like this. Okay? And so this is where your signal is off, and this is where your signal is on, right? Or vice versa, because we see that in this system, actually, when the ligand arrives, the action decreases. So this curve might be the other way around, but never mind, it's the same thing. What matters here is that you amplify this. Okay, so now the point is, suppose now your bacterium is moving in a gradient. So it's climbing up concentration. So as it moves, concentration will move this way. And eventually, if the curve is very steep, it will end up in a position where it saturates. And here it cannot respond anymore, because all receptors are on, and you cannot feel anymore the differences. So that's very bad. That's the very same mechanism that I was discussing yesterday. When you come out in the light from the dark, and then you don't see anything for a while, that's just an effect of saturation. Your visual receptors are saturated with light input, and then adaptation must occur. So what is adaptation? Is actually the fact that this curve, you can shift it sideways, right? So if you move to higher concentration, your curve follows you and will displace here. Then you get back again to lower concentration for some reason, then your curve comes with you. So you always basically want to, sure. Pardon?
So when that curve switches?
Yeah, it follows. It's not the switch, but it follows slowly the change in concentration. So yeah, right. Right. The other curve is, so this curve is what happens if there is no adaptation. Okay? But if there is adaptation, the curve shifts with the signal. So if your ambient concentration, so you suppose you are in a gradient, right? So, and this is the curve A that applies here. Okay? So you're moving here at this concentration. You're moving back and forth, and you're measuring, and you can see some difference just along one run. You see that there, before it was some concentration, then it's slightly more. Then I increase the run, and that is, I see a small difference around this concentration. So this is the concentration level. Let's call this C naught. So this is something C naught. You're here. You wiggle back and forth, your bacterium. Then you can see small differences, which are largely amplified in the response. Now, since you got the right message, you slowly go upward, you drift. Then after, say, a few minutes, you've made several tumbles, and then you're there, which corresponds to another concentration. Now, in absence of adaptation, you now are in new concentration C1, which is here. And then you cannot see differences anymore, because along one run, you will see a very small difference in response. Bacterium does the same thing. In this case, it will just go straight, run straight without ever tumbling, because it's saturated. Then what does the bacterium do? It adapts. It shifts the curve sideways, so that this curve always lags behind and tries to follow the local concentration level in order to give the strongest response. That's the sensory explanation of adaptation. We are using a sensory idea. We cannot detect differences, so my curve has to follow me. In this expression, what takes care of shifting the curve is methylation. So you see it appears here. So if you move with the ligand and methylation grows accordingly, you can maintain your activity fixed, which is adaptation. That's the basic mechanism for following up. Right?
You mean to say if I reverse the signal, if I reduce the signal in backward delta, it's a different path?
If you reduce the signal?
If I'm increasing the signal,
Yeah?
the range of signal is sharply increases.
If it sharply increases, it transiently, your activity will go high, and then it will recover the initial value. Right? If you change it suddenly, rather than going smoothly like this, you make a step in time. You are here, and then all of a sudden you find yourself here. And then this curve slowly moves, because of methylation moves this curve slowly, and then eventually you find yourself back here. If the change is large, it may take minutes. If the change is small, it takes seconds. Okay? So, and when what actually regulates the accuracy of your adaptation is the availability of methylation sites, basically. As long as you can methylate, you always adapt. But at a certain time, the methylation sites will be over. And at that time, you'll not be able to adapt anymore. That's the level where the signal is so strong that you just cannot adapt. Right? So you might lose your ability to chemotax for two reasons. One is because you run short of sensitivity. Okay? That is, your concentration goes below this K off or above this K on. Right? Then your cluster doesn't respond anymore. The signal does not even enter the cell. Okay? You cannot detect it. It's just too strong or too small. Within this range, you detect well, but then there is this other issue of adaptation. So you might lose sensitivity to signal because responds to a signal because of sensitivity or because of adaptation. The two things are independent, right? Typically, what happens is that the shortest range is given by sensitivity, and adaptation works on a much larger range. So you should care about sensitivity first. So when the methylation sites are abundant, and you have no troubles in methylating the methylate, you always find a place where to methylate or to demethylate your receptor. So you can always try to shift the curve. This means that these two prefactors, which refer to the availability of methylation sites, disappear. And then the equation, this becomes unity, this becomes unity. And then the equation becomes a very simple equation. You see that the methylation rate changes in time only as a function of activity, which means that at a steady state, the activity will not depend on the ligand. Right? That's the formal description of what adaptation is. So in the next few slides, we just discuss this. So these are the experimental data. This is the tumbling frequency, the bias that you have seen before, as a function of time for a population of bacteria that at time 0 have been submitted to an enormous increase in concentration of aspartate. So going from 0 to hundreds of micromolar. So what happens? They are just flooded with this, and the tumbling frequency goes to 0. They all start to run, right? Because what has happened is exactly what we were discussing before. We were here. All of a sudden, the concentration has shifted here. The activity has gone at the top level. In this case, it's gone to 0 because that's always reversed. But this means that the tumbling disappears and all bacteria run. And then the molecular mechanism tries to catch up. And it starts to demethylate all this. All the sites have been methylated. You have no ability to respond anymore, to adapt anymore. And you start very slowly demethylating. So liberating, freeing all these sites that were occupied by methyl groups. And this is a slow process, but you take it away, take it away, take it away. And when everything is over, then you can demethylate, demethylate, and then you can recover by demethylation the original state. And so again, the number of methylated sites is a fraction of the whole possible number of events, and then you can respond anymore. If you here were to repeat the experiment, the same thing would be reproduced. The smaller the jump, the shorter the time. Okay? And if you do this experiment, you see that different bacteria respond with different timescales to the same stimulus. So you give the same jump, you see that some of them recover in 10 minutes, some other in 5, some other in 20. And the origin of this disparity, as I said before in the previous lecture, is that the number of molecules that operate this methylation and demethylation is small. So they can change from one bacteria to the other. One may have twice the molecules, which means twice the rates. And this explains the differences in times, in the individuality of bacteria in their adaptation response. No, I wouldn't say so. I mean, it's rather the other way around. As I will discuss before, after, it's rather the environment which selects the right time scale and evolution that has matched things in order to have a biochemistry that chooses the right time scale. So the bacterium as an environment has to cope with that, and so everything adjusts along evolution in order to get the right time scale. Depends on the stimulus, as I said. They may stay there for half an hour if you just flood them with stimulus, and then slowly this demethylation, it takes forever to demethylate all the possible receptors. You have to demethylate 10,000 receptors with few molecules, 100 molecules, so one at a time must go there, take the methyl group out. The kinetics is not so fast, so it takes time, but it always recovers there. Okay, so it is interesting to point out that at the level of engineering, the mechanism by which adaptation is reached is actually very well known. But you all know how this circuit works, actually. Because I think everybody of you has used this circuit once in a lifetime. So this is the description of what happens at the level of molecular chemistry. This is just a schematic view, and this is an even more schematic view of what happens. There's an input, which is here the ligand concentration, and there's an output, which is here the concentration of KYP, which is the signal to the motor. And then here, there is the methylation level, which acts as a buffer. Now, this architecture is called an integral feedback loop. And it's actually the same way that thermostats work. The air conditioning system works exactly this way. It measures the outer temperature and then produces a feedback that sets the inner temperature at a fixed value. If this goes up, the system reacts and then produces always the same temperature. There will be transient, right? If the change in temperature outside is from 20 degrees to 40 degrees in a minute, it will take several minutes before the temperature inside the room will get back again to 20 degrees. So depending, again, on the input of the change in stimulus, and depending on change in stimulus, it will take more time to respond. But the architecture is the same. So it's a very, very simple architecture. Basically, there are just two of these architectures that can produce adaptation. One is this one, which is used in bacteria and in eukaryotic cells for receptor trafficking. Receptor recycling is the modality of adaptation for higher eukaryotes, which uses the same scheme, conceptually. Of course, the actors are different, but the scheme, conceptual scheme, is the same. And then there is another scheme for adaptation that we will discuss for the amoeba. And those two are basically the only two that you can build up with a small number of ingredients. As promised, very last thing that I want to discuss about the molecular discussion is that there is a striking connection between the behavior of these sensory systems and some known laws that have been discovered in the 19th century about our sensory ability. So this striking connection is made through what is called the Weber-Fechner law. So these are two laws that were basically are two expressions of the same concept that have been discovered in what is called psychophysics. That is, a quantitative study of how we respond to stimuli. And typically, the stimuli experience may be vision, may be the response to a weight, maybe auditory stimuli, right? So the basic idea is that is the following. Suppose you just take your hand like this, and then you put a pen over your hand. I don't know how much it weighs, but it can be in the tens of grams. Then you can clearly feel that there is a pen, right? You feel the weight difference. You feel the pressure of the pen on your hand, you feel the weight. Now suppose that you're holding something which is heavier. So you're holding in your hand some dumbbell which weighs 5 kilograms. And then you put a pen onto it. Will you feel the difference? Maybe not. Maybe we just feel a smaller effect, right? So this is the idea of just noticeable difference. You start with a given stimulus, and then you put small differences. You add something until you can feel that the difference is there. And now what this law says is that in order to feel a difference, you must add something which is proportional to the weight that you already put. So if you're holding something which weighs 10 grams, you can respond to 11 grams. If you are holding 10 kilograms, you must add 1 kilogram in order to feel the difference. 1 gram is not enough. So which actually means that we tend to respond proportionally to stimuli and not to the absolute intensity. We detect fold changes. We detect changes from 10 to 10, from 100 to 120% is okay, but this is the relative intensity of the stimulus. And the same thing actually can be rephrased by saying that the subjective sensation, that is what we feel about the weight, it's not the weight in itself, but how we feel it, is not proportional to the weight itself, but to the logarithm, right? So in this case, your stimuli, if you double the stimulus, okay, this will just respond linearly. You will feel linearly a stimulus that increases instead exponentially. Okay, these two are the same, two sides of the same coin. And now the interesting thing is that in our model, when there is adaptation, so this is the activity level, and it's a function of methylation and ligand concentration. In between these two ranges, K on and K off, you have exactly the same behavior. That is, if you take the derivative of this expression, so how it changes for small changes in the ligand concentration, you just do the calculation. It's very, very simple. And you will see that the change in activity due to a small change in concentration goes like the inverse of the concentration. And this is the activity which is a constant, actually, because you adapt. So this proportionality of the derivative to the inverse of the concentration is just this logarithmic sensing. Okay, so it's pretty striking that there is this very tiny system, molecular one, which actually displays the sort of behavior that we see in our larger organisms. And this has been explored recently very much in the literature and goes also under the name of fold change detection. And actually, there are many molecular systems that displays this ability, which sort of makes sense. You don't want to detect absolute differences, but you rather want to rely on relative difference. You want to be sure that one thing is different from the other because it's twice that value. Yeah, right. The one that appears here. It can be a one or the other. I mean, it can be any ligand, any. Are these two limits K on and K off, below which you don't respond and above which you saturate? This means that in this range of concentration, you don't shift. Yeah, that would be manipulating K on and K off, which actually you don't. But what you do is that in this range, you respond to fold changes. But these are two independent things. You can have adaptation without fold change detection, and you can have fold change detection without adaptation. The two things, in general, conceptually don't go together. But in this case, they are together. Okay, so that was basically all I wanted to do and to say about our long descent from the large scales to the small scales. And now I will discuss how to connect the levels from two very different viewpoints. Just for reasons of time, I will be rather rapid on this, but you can interrupt me at any time if you're missing something. So the first thing is that since we have all this deal of information, all this amount of information about all these different levels work, it is actually legitimate to ask whether we can look inside the cell without actually measuring anything molecular. So can we, from the behavior, the trajectory, infer what is happening at the molecular level? This is pretty much the take that that a psychologist would have, right? So you feel that something is not wrong, you go talk to him, he makes questions, you give answers, right? Nobody is touching your head at any time, okay? And then you get some insight on what is happening in your brain, so I better put you on some drug that might help you feel better, right? So this is based on behavior, you make an inference on what the molecular mechanism inside your head might be going wrong. Or just the MD that visits you, it's the same thing, right? He visits you, asks you about your behavior, and then infers that something is going wrong in your kidney, right? Without actually cutting you in pieces and looking at your kidney. That's the surgeon that does this, right? So in a sense, the molecular biologist acts like a neurosurgeon, right? And this is just like I can as psychologists, looking at behavior and try to infer the results. Actually, the nice thing is that you can do this. Once you are equipped with your model, which you know it works very well, the technical physical question that you ask is, can I infer the parameters of my model, so all the rates, the intensity, the sensitivity, from the knowledge of the trajectories? So suppose I collect a bunch of trajectories, or as a matter of fact, you can do this with one trajectory, single trajectory, but provided it's sufficiently long. Can I, from this single trajectory, infer what are the biochemical parameters? Well, you cannot do for them all. That's the first thing I want to say as a disclaimer. Because already in this simple model, when there are just a handful of parameters, just from trajectory, you cannot deduce them all. You should combine different experiments, subject them to different stimuli, and then in that case, you might combine them all. But if you make just one single experiment and you let bacteria run up a gradient, you can infer exactly the intensity of the response and the time scale of the response for a single bacterium. Right? You can do your bacteria psychology in this way. And now I'm cutting a long story short because all of this requires some knowledge of how these inference methods work, but it's nothing really extra technical. I just wanted to get you the message that once you have a model that helps you connect the level, then you can go from one level to another with agility. And then if you use this approach to measure indirectly the properties of the response, you find something very interesting. And here I'm showing the results. So if you look at this now, it's all written in parameters that make sense once one writes the model, but I will explain you what these pictures are. So forget about the first one, just focus on this one. So this curve is actually related to the response function that we have seen the last time. So how the bacterium responds to a spike in concentration. And this parameter gives you the precision of adaptation. So how well you will get back to the initial level after having changed this. So you see that all these points are individual bacteria which are moving in a gradient of methyl aspartate, which is just aspartate, but it's not metabolizable. And you see that all of them basically fall in 1% precision of adaptation. Just every single bacterium wants to adapt very well. But now if you look at the intensity of the response, the sensitivity, or the time scale, you see that the hell breaks loose. Every bacterium basically chooses a different time scale, a different intensity of the response. There does not seem to be specific pressure on one time scale, one intensity of the response. This, again, is certainly related to the fact that the copy numbers of your proteins inside the cell, the copy numbers of the amplification machinery is not so high. So there can be individual variation. The mechanism for this is clear. What is less obvious is why. What for? What is the functional value of adapting so well? Is there a reason why bacteria are just widespread in behavior at the level of time scales and intensity? So we will discuss now in
in greater detail the issue of adaptation, which is probably the most striking one. Because every single bacterium here seems to be very keen on being adapted to this signal. As far as this behavior is concerned, so there is a sort of conceptual explanation for this. And this conceptual explanation is called bed hedging. So it's a strategy that you use when you don't know what you're going to face. You're going to face an environment that changes a lot. So you don't want to put all your eggs in one single basket. You want just spread your probability of facing your ability. Remember, all these bacteria have the same genome. They're isogenic. They just differ by their behavior. So the population gets there. Then there is an environment which is with very weak gradients and very slowly changing. And then this bacteria which responds with high sensitivity and with slow timescales will be okay. But then if the environment suddenly changes to a situation where the gradients are strong, but they change very fast in time, there will be other bacteria that will be better off. And in your population, you always conserve enough diversity to cope with different environments. So that's the basic qualitative explanation. Because in order to make it quantitative, you should show that exactly this sort of variance, so this sort of spread of the population, is the right one to deal with this environment. Because this actually explains everything, because it doesn't tell you why the intensity of fluctuation is, why it's 1 decade of variation in the sensitivity. Why 10 or not 2?
So this refers to the fact that you have broader and sharper.
No, not quite. This refers to the fact that you can have shallower or steeper. Yeah, right. But sorry, I was messing up. Because actually I thought the ends are fixed. It's the slope that may change. So you may amplify less and that's what is in here. And what is in there is the fact how quickly you shift in this direction. So maybe I was unclear before. All right, so let's say that this thing, we basically understand it qualitatively. Of course, there's a lot that remains to be done in order to understand it quantitatively. Here there is a connection with a branch of mathematics. I would say that there is mathematics, that is game theory that waits to be applied to this sort of problems, right? Sure.
Does the property for a cell remains constant, or it also keeps varying?
Along one cell cycle and even more, it stays the same. So the turnover time for these proteins is pretty long. They are quite stable. So in the, say, 1 hour typical time scale for doubling, they may be faster or slower depending on nutrient. These basically stay where they are. So they then don't change as fast as the environment does.
So it can easily be said as a property of population and not the individual adapting it.
Well, so which one are you referring to then? Each individual adapts. Okay, but each individual has its own favorite response. Is that? Okay, so adaptation. Now, to add some more confusion, which I hope I will be able to dissipate in a second, now I'm showing results about adaptation. So the same picture is here, but for different chemicals. Okay, this was for aspartate, but you know the cell senses other chemicals. So let's go and look what other chemicals do. And then, here what you see. So aspartate, the previous data are the black dots. So they adapt pretty well, right? It's a larger scale now, but you see they fall within this 1–2% precision of adaptation. Glucose, the cyan curves, okay, that's pretty well, too. Now this is a mutant which lacks all the machinery for adaptation. And of course it doesn't adapt anymore. It fails because it has no way to do that. It also responds to larger concentration because it's also less sensitive, but is an independent fact. So the curve is shifted here with respect to here. And now serine. You know that serine is favorite E. coli food. And then you see that in some concentration ranges, at least, there's no adaptation to serine. Leucine, a repellent, no adaptation to leucine. And then you start asking big questions. So why? Why is all that? So I was reasoning so beautifully here about this is something that serves me to sense better, but this applies to everything. It doesn't need to be something which is specific to aspartate or to serine. So what's changing here? Why? Why would some, yeah, sure. Sorry? Yeah, right. This is a repellent, so the response is reversed. It's negative. So everything is flipped the other way around, but here want to go down, the gradient. The one go where the noxious substance is, such as leucine. Yes. Okay, so in order to answer to this question, let's focus on an even narrower question first, but which is of great interest. And the question is, what is the value of adapting? This is one explanation. It serves you to sense better. Then this casts doubts on explanation. Then you want to give an answer. Why on earth should I adapt? What do I gain? Sensing is okay, but bacteria do not live for sensing. They are not sensing machines. They use sensing for some purpose. That is, doubling themselves. So what is the value of adaptation in that sense, in the evolutionary sense? Let's try to give a rational answer to this question. That is about the function of chemotaxis. Now, a very important insight into this question actually was given some years ago by Pierre-Gilles de Gennes, who asked the very, very simple question that actually never dawned on other people's mind before. So let's assume that there is a linear gradient. There is bacterium into the linear gradient. Let me ask a very simple question. Which is the best response for the bacterium? The one that makes me go up faster? That's a pretty obvious question. And actually, this question motivated, since bacteria have chosen this sort of movement, and they live into this gradient, that their machinery must have optimized their ability to go upwards. That's what selection does. Then you just try, okay? Or you do some calculation and you try to optimize. That is, find the one that produces the largest drift. What is the response that produces the largest drift? And what you find is that the response is like this. It's so positive. This is the response to a spike. You remember, the observed one goes up, then goes down, which is the part that pertains to adaptation. This then goes up again. This does never go down, which means that this does not adapt. This is a response function with a single lobe, positive lobe, never adapts. When you are into a gradient like this, and you care about eating more, not being very keen on sensing what the gradient is, because bacteria don't ask themselves, well, do you think we're living in 1 micromolar per millimeter? Just bacteria care about going and eating. And the best thing that they can do is choose this response, which is not the one that you observe. Then other questions came. So maybe this is not the typical environment that bacteria see, after all. We've always been thinking of the gradient, but that's our input to the problem. So maybe what bacteria feel typically are just small patches.
What is it that bacteria do in response to this?
It goes upward, right? But it goes faster in this case rather than with other response. The behavior is the same, but only the drift is larger. And what happens is that while going upward, it even accelerates, actually. In this range, always in this range of concentration. So suppose let's compare. This is the bacterium which adapts. It will go, okay, it will just go like sort of rectilinear motion on average, upward, if it adapts. Velocity is constant. If it doesn't adapt, it will actually accelerate. because as you see new concentration, right, it also adds another excitation to its motion. So I should go through a little set of steps in order to show this, but maybe with the next step. But what you can show is that actually, even at the level of a single run, this goes faster. So you could show it on different scales, and it's all consistent. Just because I mean, you don't. When you adapt, there will be a phase in which you tumble more, and this is just losing time for you. It's not effective in terms of going up gradient. You're losing time to measure to better measure the gradient, but this doesn't help you. To a pulse, the response function. A single pulse, very sharp delta function. Yeah, when you make a convolution of this, the response that you get goes up and never goes down again. So what if the environment is a static patch? It's something that stays there. So the algae that has gone dead and emits for some time your attractant, and then all bacteria wants to go there and eat the food. Then if the patch is sufficiently long-lived, the response that you get is you better be with a negative lobe. Well, that's completely counterintuitive. This just tells you you should go away from the gradient. Well, that's silly. But not so silly, actually. Because if you happen to be on top of the patch, then you will stay there, and you will exploit this patch better with this response. Okay? So if the resources are staying there for a long time, you better switch to a strategy where you go there and you happen to be there by chance, and then when you're there, but you stay. Because the patch is there, and you won't exploit it all. So, of course, then people start to think, okay, maybe the response is some compromise between these two. But this is hard to defend as a point, because in order to get perfect adaptation by combining these two responses, you need to do some fine-tuning. So the two constants in front of the response must match exactly in order to have an adaptation. Right? It should be a very fine-tuned compromise to get that specific response. So it's not convincing that it's sort of seeing some of these environments and some of the others, because the proportion in which it sees one and the other must be very precisely specified. So shouldn't we just looking for an entirely different explanation of adaptation for this problem? So, and first of all, which cares me the most in this context of a school, how can one approach this problem systematically? Now, I'll briefly give you the conceptual sequence of the ideas that come with this problem, and then we get to the answer. So everything must be recast in some way that connects the response, how bacteria sense the environment, to their fitness, how quickly they reproduce. Let's go from the top to the bottom. Let's start from the fitness. So what is the main determinant of the fitness? Fitness is just ability of reproducing. For a bacterium which undergoes just duplication, basically it's a measure of the time that it takes to you to replicate. If you replicate fast, you're better off. If you replicate slow, you're in a bad condition or competition with the others. So what is the determinant that makes you grow faster? Nutrient availability, right? So when the nutrient is abundant, all bacteria basically grow with the same rate. They eat all that they can. But when the nutrients become to be limiting, that is small enough, then your growth rate depends crucially on the level of concentration. So this is the first ingredient that you put. My fitness will be a function of the uptake. So how much nutrient can I get along my lifetime? Because that's what determines the time of growth. Then you make the next step. You want to connect how much you input as nutrient with the properties of your trajectory, right? So given a distribution of nutrient, and given a trajectory, how much will I get? And this is formally written like this. How much I get is in general a function, which might be very complicated, of the trajectory I take and the distribution in space and time of my chemoattractant, which is a proxy for my nutrient. Okay? I look for chemoattractant, but actually what I look is for nutrient. And then final step, you want to write your trajectory as a function of the response and the chemoattractant field. Okay? So now we can climb it up in order to check if everything was right. So the trajectory that I actually take depends on how I respond to the external stimuli and how these external stimuli are arranged in space and time. Once the trajectory is given, it moves around and collects food along the way. And the amount of collected food is a function of how I move and how much and how the food is distributed. And finally, I can go from uptake to fitness. And the last step actually is not very important. Basically, every growing convex function of the uptake will do for our considerations. So we will basically stick to these two steps, which are challenging enough. How can one do this systematically? So the basic idea is that we start with a model which describes the microscopic behavior of bacteria. So it just describes that it just tells that they do run and tumble, and that the duration of the runs is modulated by the response function, which is in itself linked to the molecular aspects of the problem, right? So we just build up a model, analytical model. We write down the equation. It's master equations, typically, in continuum space, of course, which tell you the probability that your bacterium will be at a certain point in time with a certain velocity, with a certain concentration of KYP, with a certain activity of the receptor. You have a list of internal variables and a list of velocity and position that specify the state of the bacterium. And then it proceeds and it feels rotational diffusion. There is tumbling from time to time according to the prescribed rates. So you build up a model which describes what you see, right? But at the microscopic level, the level of the single cell behavior, and includes information about the molecular response. And then your goal is to go from this model to a macroscopic one. Because at the macroscopic level, you will be able to write down equation for the uptake. Right? And the tool to do this is you exploit the fact that runs are short in a sense. They are few microns, 30 microns long. They are 1 second long. And then your concentration field, because of diffusion, will vary on longer length scale and on larger time scales than this. This is essential to this process. It's scale separation. There are two different scales in times in the system, the scale of this random motion and the scale of variation of the concentration field, which are separated. And this allows you to make the jump from microscopic to macroscopic. And it's basically the same thing that you do when you go from kinetics to hydrodynamics, from mean free paths to viscosity. And we're doing the basic same thing, but for bacterial motion. This goes through some technical steps, which I will not discuss, but basically are reasonable assumptions. One important assumption is that you can do all this in the linear response regime. The non-linear response is a pain in the neck. But the linear response regime, which is pretty large, you can deal with. And you use scale separation, you get to hydrodynamic extraction. How do they look like? Surprise of the surprises. They look like the Patlak-Segel-Keller model. Only that we have derived microscopically now. So we have an equation for the number density of bacteria, or if you wish, the probability of finding a bacterium in a certain point of space, which is the same in a population of non-interacting individuals. It has a chemotaxis term with a coefficient chi. Remember, everything is linear, so we won't have these cumbersome functions with fractions, powers, etc. But the structure is the same. There will be a diffusion term, and there will be this diffusion coefficient actually still bears a dependence on concentration here inside. So there are two parameters here. One is the chemotactic coefficient, and this one is what we call a chemokinetic coefficient, because it just tells you that if the concentration is large, the diffusion coefficient effective will be larger. It means that bacteria sort of make an agitated motion in presence of higher concentrations. Now, the upshot of all this, we actually re-derived sort of macroscopic equation starting from microscopic, and the advantage is that these two coefficients are now related to the molecular response. These are functions of the microscopic behavior of the bacterium. And their explicit expression are given here. There are parameters which are related to the properties of motion. Never mind about that. The important thing is that you see they make appear the response function. This k of t is this response function that I've been talking all the way around. Response to a pulse. So you have this chemokinetic coefficient. Very good. And then there is this other coefficient, gamma, which is equal to the integral of the response function. And now you see, suddenly, adaptation pops out spontaneously. Because this quantity, when you adapt perfectly, is 0. Because this being 0 is the requirement to adapt perfectly. So you clearly see in your effective equation, adaptation stands up immediately. You can see this is due to adaptation. And this is due to chemotaxis itself. Then you can go to the next step. And then you can write down equations for the uptake. So now that you are in this setup, it's actually a relatively simple exercise, this second one. And what you do is just, you say, okay, I'm putting myself in conditions where the nutrient is limiting, the concentration is low. So the influx of food will be regulated by diffusion, as we started. So how many molecules I get in the unit time will be essentially proportional to the concentration around me in the environment. So this means that the total uptake that you get, if you wish, is the total uptake now over the population of bacteria, will be the integral over space and the integral over time of the bacteria that eat and the concentration that they feel. It's very simple. This is just the consumption of bacteria all over the times and the space that I'm considering. Then if you use this simple expression, then you come up with another expression that now makes appear explicitly the responses. So you just solve for this equation and plug in, and then you get an expression for the uptake. Okay? I don't expect you to follow all the details of the calculation, just the idea that's behind. Using hydrodynamic equation helps you write down an explicit functional quadratic of the concentration. So you can relate, given a response and given a concentration, you end up with an uptake. And then if you do that, the result is a very complicated form, but it's actually very easy to interpret. There is one part of the uptake which you cannot optimize because it doesn't depend on your response. So this is what you will get even if you have no chemotaxis signaling. It's just being at the right time at the right place. Whatever this means inside, that's the meaning of the term. And then there is this other contribution, which actually you see depends linearly on these coefficients, and these coefficients in turn depend on the response. And this is the part that you might wish to optimize. And then you see it makes appear the gradient and the Laplacian of concentration, so local properties of the concentration field along the trajectory. And now this is enough to understand everything. Because you will notice that this term, which is sort of the gradient that is experienced by the bacterium along its trajectory, is always positive because it's a square. Whereas this term may be positive or negative depending on where you are. If you are on top of a hill, you're negative. If you are on the bottom of a valley, you're positive. It depends. And this term, which changes sign, is proportional to adaptation. And then you get to the conclusion that what adaptation does is it just kills you this term which may make you positive or negative. And as a result, if you adapt, you are sure that you will gain something. Because your gain, your added value due to the response will always be positive. So that's a very important message. It's not that the bacterium does what it's best in that specific environment. It does something that in every environment would give you a little amount of gain. It's a sort of very, very conservative strategy, risk-averse strategy. You want to always get something out of it. Maybe it's not the specialized response. There are other bacteria which are more specialized, will do better in that environment. But then if I change environment, I will do good as well, and they can fail miserably. So that's something which is very, very akin to actually behavior which is well known in economics. These are what are called minimax or maximin strategies. These are strategies that optimize the gain in the worst possible case. And these are strategies that always warrants you as a response to strongly unpredictable environment. Because in that case, you can cope well with all these different environments without having a specialized response. Conversely, if the environment is repeatable, predictable, it's always the same, like a gradient which is always there, then you better switch to a specialist strategy. Say the positive lobe, which is the best one in this case. So this is all graphically actually summarized in a picture, which gives you the uptake, the gain that the bacterium has given a response and given a configuration of the external concentration field, right? So if you are in the bottom of a well, you better respond with a positive gradient because it takes you rapidly away. So you're very good at this, very good uptake with this response. If your response is mostly negative, you will do very bad because you're here. You're staying here. The situation is opposite if you are on top of the hill. And then the adaptation is just at the center point of this landscape. It sits here. And it's the place where basically you see you can do well in all possible situations. So that's the basic take-home message for this part. Maybe adaptation has not to be understood just like a sensory mechanism, but as a mechanism that actually produces fitness in an environment that changes unpredictably, and it's actually very hostile. It does the worst for you. Okay? Incidentally, if you use this theory as a prediction, so if you impose that your best response would be this minimax response, you come up with a prediction for the response function. And then you can compare it with experiments, and then you see that you get the right timescales. Okay, so this theory, besides being qualitative, has also some quantitative implication. Of course, it's a linear theory, so one thing that you will never get is the amplitude of the response, because everything rescales. So you have to fit the amplitude, but you do not have to fit these values. Actually, what determines the timescale, not surprisingly, is the rotational diffusivity, which again, as I said, sets the main timescale for the responses. Because above this length, this timescale, you cannot respond effectively. So we are basically done with bacteria. Last slide is just to remember that there are many senses besides chemosensing, and many of them are very important for bacteria already. Bacteria can sense temperature gradients because they favor our gut temperature of 37 degrees. So they respond to temperature gradients by collecting themselves in bands at 37 degrees. So this is low temperature, high temperature, and all bacteria go there. And as I said, they use the same molecular architecture serine and aspartate receptor to do this movement as well. Bacteria can respond to light. They actually have proteins, receptors that are called bacterial rhodopsins, which are the equivalent of our rhodopsins, except they are 7-pass transmembrane proteins, but they do not have G protein coupled part inside. But they are sort of precursors of the receptors that we have in our retina. They can respond to oxygen, of course. Oxygen is very good. Some bacteria can respond to the magnetic field. Okay, so they have vesicles which are filled with magnetic beads, which help them align with the Earth magnetic field. And if you wonder why should a bacterium want to go north or south, well, actually, that's not quite its interest. The point is that the magnetic field lines are not only oriented from north to south, but also they tend to go inside the Earth. So they use this as a mechanism to distinguish up and down rather than north and south, which is not particularly helpful, being able to orienteer your map on such a small time scale, source. And then there is also galvanotaxis, that is the ability to sense electric currents, which may be related to pH availability, nobody knows. So there are many, many senses already at the bacterial level. And of course, the level of understanding and description of all this is not as evolved as the one that we have for chemotaxis. But these are relevant mechanisms for their physiology and response. Okay, now we take the next step, and we go to the higher step in the ladder, which is eukaryotic cells, single cells. So I understand that you, Professor Cox, should have lectured on dictyostelium, right? So you probably familiar with this picture. So dictyostelium has this very interesting life cycle. When it goes from an amoeboid isolated life form, everyone stands for himself. When it's starved, it starts to aggregate and switches to a facultative multicellular behavior, in which even there are cells that start to differentiate and behave as stalk cell or as spore cells. So it's a very complicated social behavior, with extremely interesting. I would not touch upon this at all. I will be mostly focusing on the aggregation, that is the chemotaxis aspect. So in order to aggregate, some cells start to emit this cyclic AMP signaling molecule, which is naturally produced when cells start to starve. So they emit this chemical, and then waves of aggregation start to form and bring all the neighboring amoebas close to the center when they start to form a mound, and then a slug, and then etc. This is a very interesting process, which actually, again, as previously, has first been described at the level of population. So you can write down an equation for the number density of amoeba in a certain point of time, and then you see how this evolves in time. Does it explain what I see? And you quickly come to the conclusion that if you put the right ingredients, your system starts to behave like what is called in physics a self-excitable system, which produces waves of its own, responds to these waves, and this is a good description at the population level. Of course, we cannot go at all through the details of this, but the spirit is the same. Even the equations are different, the mechanisms are different. The spirit is the same as building up a Keller-Segel equation, right? Same kind of approach. Then we go to the cellular level. These amoebas you see, they are sized about 20 microns, typically. Okay? So they are definitely larger than bacteria, but not so large so that their environment is completely different from the one experienced by bacteria. So diffusion is still the major force acting in carrying nutrients to the cell. These bacteria don't swim. They can swim, actually, but their typical behavior, don't swim, they crawl. So they are on a rigid substrate. They live on the soil, typically. They're on a rigid substrate, and they crawl to move. And the second important distinction is that they have a sense of having a head and a tail at variance with bacteria. So these cells are polarized, and there's a head, a very well-defined head, and a very definite front and a very different tail. So we see in this short movie one of these cells crawling. You see it emits these pseudopods, right? And then the tail follows. Here there is a gradient of cyclic AMP in this direction. You see the motion is not exactly deterministic. There is a sort of slight random component because these pseudopods are emitted sort of a random fashion, but it's much directed, much more directed than the behavior of a single bacterium, right? In red, it is actin that is labeled. Now, actin is one of the important molecules that govern cell motility and cytokinesis, that is movement of everything that is inside the cell. So what happens in a sketchy way is that actin pushes the membrane on the front, and myosin, which is in red, retracts the membrane in the back. So there are two different mechanisms. In the front, pushing of actin via polymerization, and in the back, retraction of actin cables by myosin, and then a continuous recycling of this thing goes on. It's a very complicated system, very interesting. People devoting entire careers to understanding all this, how this works at the level of motility. And it's very relevant because this sort of motility also, we find the basic features also in our cells, in higher metazoans. Pardon? Actin is spanned over all the cell, but it's preferentially concentrated in the front. And myosin is preferentially concentrated in the back. This reflects an organization inside the cell where the front and the back are chemically different, biochemically different.
So, but one of the sudden the red region disappears and appears in the other region. What's happening there?
You saw this in the movie? We'll see it again together because I didn't notice. Okay, so this is all supported. So you see, there's some myosin here as well. Yeah, right. That's what. Where is he?
That red region occasionally disappears from one region and it appears.
The pseudopod. Ah, yeah. You continually produce them and retract them. It's sort of an exploration mechanism.
It's a bundle of actin filaments.
Yeah, they continuously assemble and disassemble. It's a dynamic process. I'm making it very simple, but the idea is that you produce, you push, and then you retract. Everything goes on dynamically. That typically there's more actin in the front and more myosin in the back, but there's a turnover, of course. So the first question is, how is this spatial organization, organization realized at the molecular level? Is it a consequence of cells sensing the gradient in the front and the back? The answer is yes. All this follows initially from sensing. So the basic pathway for chemotaxis is actually very interesting from our viewpoint because it shares many interesting points with our own signaling pathways. I will describe it very, very quickly. So the first interesting thing is that the receptor for cyclic AMP is a G-protein coupled receptor. So it's the same sort of receptor that we have for olfaction, etc. Many senses governed by G-protein coupled receptors. Second very important thing is that as soon as the signal gets in the cell, it's processed through the RAS protein. This RAS protein is actually the same protein, within minor differences, that we have, and that's responsible for, I don't know, 50% of the cancers. No, it's not responsible. It's present in 50%. Mutations of this protein are present in 50% of cancers. Right? It's basically the same thing. It's really very few mutations. So this way of transducing the signal using RAS proteins is very, very old, actually. And then through this switch, because RAS can exist in two states, one with GTP bound in the active state and one other GDP bound, the inactive state. So this is sort of an equivalent of the phosphorylation, dephosphorylation way. And this is achieved by two enzymes that switch the balance in favor of being active or inactive. And This is called GAF, guanine exchange factor, guanosine exchange factor, and GTPase activating protein, which shift the equilibrium in one direction or the other. And that's depicted in more clarity in this sense. And you see that when there is a mutation, typically there's one point mutation which is particularly well known at a specific residue, which I don't remember at the time, but this single point mutation can block this cycle and let your protein stuck in an ever signaling way. Always active, always signaling. The cell is deceived into believing that there is some activating message from the outside, and then the hell breaks loose, and this starts all sort of proliferation mechanisms through the ERK pathway, MAP kinases and ERK pathway. So if you do the same thing in dictyostelium, you're actually producing a small cancer model, organism model. You can enable constitutive activity of this RAS protein, and you see that dictyostelium starts behaving like crazy, and it's very active in a regular manner. It doesn't chemotax at all, just frantically moves from one side to another, and displays a behavior which is resemblant of the fibroblast-like behavior of cancer-like cells. So this way of processing information through switches is actually a very, very common theme in higher eukaryotes. Just discussing dictyostelium, actually, the cyclic AMP signal goes through many of these switches. You see, this is just the top part of the biochemical pathway, and this is the RAS switch, but there's another one which involves this other protein, phospholipase, which is again a switch, on and off, and then another one which switches on and off some molecules that are present in the membrane and depend on other molecules which are themselves as well present in our organism and themselves as well involved in cancer. Mutations of these proteins are very important. This is a tumor suppressor gene in ourselves. This is an oncogene. Okay, so these are pathways that seems to be extremely well conserved for signaling from amoebas to ourselves. And then you see that in response to a signal, so here we put in a pipette of this cyclic AMP close to one dictyostelium, and you clearly see that there is an accumulation of RAS close to the tip. So already at the level of RAS, at this early level, the protein knows what's the front and what's the back. It's important to notice that the receptors are evenly distributed over the surface. So it's not that the receptors are in the front because the response is the reason because the response is in the front. Receptors are everywhere. They are excited by the gradient, but then the response at the level of RAS is already largely polarized in the front. Okay. So these organs already have a way to organize themselves in space given a spatial cue. And this amplification of the gradient, which may be very small, is very effective. Okay, I have to go rather quickly through this, but the main take-home message is that these switches, which have been studied in detail in the last years, provide a very powerful mechanism for obtaining amplification of the signal by themselves. So by properly arranging the biochemical constants that are present in the switch, and especially if you go to the limit where these two enzymes that turn your protein from active to inactive and vice versa, if they act at saturation, so that means that they act independently of the substrate availability, your response curve is like this. It's very steep. So you can obtain a very strong amplification by a different mechanism from the one that is used in allosteric models. But the result is the same. These switches can be used to amplify very much your incoming signal and to get a strong response out of a small gradient. Not only that, the same mechanism can be used to obtain adaptation as well, but by a different architecture from those of the bacteria. So this is again the switch. There is this protein, RAS, which may be on and off, and it's turned on by an activator and it's turned off by an inhibitor. And these two activators and inhibitors are actually both responding to the stimulus. So you see the architecture is different here. There is the stimulus, which excites two enzymes, both of them, which are actually doing opposite things. So this is a sort of incoherent behavior, and in fact this sort of scheme is called an incoherent feed-forward loop in the terminology of circuits. It's a signal that splits itself into two parts. One part that say act, another one is be put. So how can a signal emerge from this? Well, simple, it's a question of timescales. Because you first say be active, and then you say inactivate yourself at the second time. So as a result, the activator starts earlier, the inhibitor is longer, and the response goes up and then goes down. And then if you're careful enough, again, with the kinetics of these activations, in this case it must be first order kinetics, but this just means that there are some large sets of parameters where this system can achieve perfect adaptation as well. So in a single circuit you get amplification and perfect adaptation at the same time. In bacteria you had to combine different things, methylation and allosteric clusters. Here you get this for free with one single. Actually, what the amoeba does is that it amplifies in successive steps using different schemes like this. But in principle, you could have done this in a single step. And these switches are everywhere in our cell signaling cells. Everywhere. And biochemically very similar. So that when you do the same thing that you did for bacteria, just computing the level of excitation of a certain molecule of RAS, the level of RAS GTP bound at different concentrations, you see that it's always the same, which is the meaning that the system is adapted. It doesn't depend, the level of RAS GTP inside the protein does not depend on how much ligand there is outside the cell. One very interesting aspect of dictyostelium is that you might ask, is this mechanism of having a head and a tail coupled to the elongated form of the cell, that is the distribution of actin and myosin? So are these all coupled together, or does this orientation mechanism come in two steps? And there is a very simple and elegant experiment to show that actually these are two different things, and that the second one, actin organization, follows from the first one, sensing. That you just give a drug, a chemical, which is called latroncolin, which suppresses actin polymerization. And if you do that, your cells lose adhesion with the ground and become nice spheres. There's no actin anymore polymerized. The cell becomes just like a bubble. And then you can do the same thing and put some attractant with a pipette and see, will the cell chemicals, will RAS be concentrated in the direction of the attractant? Will the cell organize spatially without making use of actin? And the response is yes. And this is a very nice picture that I don't know if you can see properly. Maybe it's too dark, but the idea here is there is a source of fluorescent cyclic AMP, which is your attractant. And here there are all cells which are spherical. And maybe you can see all these crescents here. are distribution of proteins which are polarized in the direction of the cell. Which means actually that cells have a compass, have a sense of space which doesn't depend on their motility ability. So in these cells already we have a sensory part which you can decouple from the motory part. They don't move, they just stay there. If you move your source, all these crescents will move accordingly, will follow, but the cells won't move at all. They're just sensing, purely instruments. Then you might go on, and then you ask yourself, oh, can we build up a model of how this spatial distribution is set inside the cell? And of course you can, and it's a very simple model actually, which is based on the idea that the protein that does the activation and the protein that does the inhibition have a very different behavior at the spatial level. So the activator diffuses very little, so stays confined in a small region of the cell, which I tried to depict with this crescent, dark blue crescent here, whereas the inhibitor is able to diffuse inside the cell much more. So what is the effect of this? The effect that you can write down, of course, in this case also differential equation, whatever, but I just want to give you the idea of the mechanism. The idea is that given that the inhibitor is evenly spread, but the excitator is just, the excitatory molecule, the activator is essentially confined where the stimulus is, so in the front of the cell. In the front, you will have that the activator wins over the inhibitor. And in the back, you will have that the inhibitor wins over the activator. And this breaks immediately your symmetry, and then you can amplify this signal and polarize your cell in a given direction. So this is what is called the local excitation global inhibition model. So this was a very, very quick overview just to show that you have to slightly change your vocabulary. There are maybe different proteins, there may be different mechanisms, including space. But basically, you can go through the same steps. So starting macroscopic, deriving macroscopic description, investigating a level of single cell, writing down differential equation at the level of signal pathway, and then trying to combine everything together. Right? And this seems to be a good plan to deal with sensory systems in single cell organisms. Then what can, sure.
So fair to say that because of the size here being 20 microns, compared to E. coli being about 1 or 2 microns, gradients inside the cell, are maintainable and have meaning. This kind of machinery would not have been possible inside the...
Would have been more difficult. It's possible to have something similar in bacteria which are rather long, which may happen. But I totally agree with the argument. Size plays an important role here. And it also plays an important role, the fact that the cytoplasm is also more viscous here, and it also impairs diffusion. And there are also other degrees that add to this, because you can have molecules that attach to the membrane, and so this is another way of localizing, compartmentalizing, using membranes, which is in E. coli, in bacteria is much less used as a way to produce localized distribution of chemicals. So then we take the big leap in the last 15 minutes, and we move to something that starts being really close to ourselves. And it's not the mice, but it's the insects. Okay? It's close enough, as long as we focus on olfaction, as you will see. So olfaction is the chemosensing, the chemical sense at the level of higher organisms. Basically, all metazoans communicate very much through odor signals. All these signals are called semiochemicals, which means chemicals that carry a message. And they are heavily used in communication. One generic name for these semiochemicals is pheromones. The pheromones just means that are hormones, that is substance that stimulate But also there is this prefix which comes from the Greek pherein, which means to carry. So these are signals that, at variance with hormones which work inside our body and communicate from one organ to another, these are emitted outside the organism and help carrying other organisms, typically close to others or away from others, and they are carried by the environment. So how might have this olfactory communication have evolved? Actually, the hypothesis is very simple. It is believed, and there are some evidences, that the mechanism is actually the following. Naturally, every organism just delivers in the environment some metabolites, some residues